Welcome to Systems Biology of Aging. My name is Alon, and I always like to start my classes with a song. This course has a theme song because it may seem unprofessional, but in fact, when you smile and you breathe, you live longer. Aging, aging, aging here and there. Aging, 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 aging everywhere. Now aging is a song that is universally sung. Aging is a thing you can ignore when you are young. But when you are older and begin to have dementia, you'll think back upon this course and wish you'd paid attention. Aging, aging, aging here and there. Aging, 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 aging everywhere. So I'm very excited and happy to start teaching with you. When I teach, I like to write down here on the right side the topics that I want to teach. So welcome, I said, and song, right? So we're already making good progress. I want to dive right into our first physiological feedback loop, okay? So our mind could be in a relaxed state, which is really good for learning and listening and memory. And when we're in a relaxed state, our body does something, which is our breath is different. Our breaths are long, like long, slow breaths. But being human beings, we can also decide to take a nice deep breath, which actually, if the inhale is short and the exhale is long, relaxes our autonomous nervous system. It's good for the sympathetic nervous system, and that opens our ability to hear, listen, and remember. Right? So in this course, we're going to practice taking nice deep sighs of relief from time to time. You don't have to, but if you do, I promise it'll help you feel good. So I'm going to demonstrate. It looks like this. Right, so let's all together take a nice deep sigh of relief. Great. All right, so that's relaxed breath. Now, since we're starting a course, it's very important for me as the teacher to do the frame setting, because we're going to the unknown together. So we need to set a good frame, which is topic, target, times, rules, and roles. So what are we doing here? Where are we going? So the topic is aging and longevity, systems biology of aging. The target is by the end of this course, you'll know the cutting edge of the research field that tries to understand biological aging and extend the human health span. You'll be able to build simple mathematical models that capture physiological questions that are fascinating, like what's the rate of aging of diseases, what happens if we do an intervention in midlife at a certain age, what effect it has. And so it'll be for basic science curiosity, understanding one of the big unsolved problems in biology. We know a lot of how we develop from egg to embryo to body plan, not a lot about the other side of life. So this is a huge cutting edge, very, very field with huge momentum right now. My goal at the end of this course is that you guys will feel you can do something in this field and want to work on it. We'll see. So that's our target. The time frame is we'll have a class once a week from 2:15 to 4. And roles, so you have to be yourself and ask questions. We have a special role, Ben Shinhal, can you get up? This is Ben, he's a teaching assistant. He's there for you, to support you. This is his email, and he is going to advertise office hours and also do a tilgul, which is a frontal. And during a week while I'm away abroad, Ben will fill in for me. My role is to try to be as clear as possible. Rules. We're going to have, how many of you are taking this for credit? How many of you are just auditing? Good. Welcome. Those of you who are taking it for credit, we're going to have an exercise every 2 weeks. These exercises will be, we'll have a little bit of solving mathematical equations, a little bit of programming, stuff like simulating, a little bit maybe of tasting big data. And then we're going to have every 2 weeks. And then we'll have a final exercise, not an exam. And all of these are pass-fail. So the whole course is pass-fail. And the reason I do that is to, again, make you feel relaxed and have your motivation be intrinsic as much as you want to study. The exercises and the final exercise and the exercises and the final exercise can be in pairs. And especially I like pairs from two different disciplines. Doesn't have to be, but... and the exercise in the end, you'll see, it can be very creative. It's to choose a topic that has to do with aging, longevity, and address it with the tools of the course. And it could be anything, because aging is actually the human condition, or the yeast condition, or the worm condition, whatever you'd like to study. So usually you get very creative final exercises. Any questions about the structure? I'll pause till there's a question. Yeah.
Will there be songs every class?
Yeah, will there be songs? Yeah, I'm going to do the theme song every class, and then it's a new course, right? So you're the first, it's like you're the pioneers. And because it's a really young field in certain ways. And I want to write extra songs too for different topics. So I write with my daughters, by the way. They're really good at it. Thanks for asking. Okay, so we have the frame setting, the topic, target, time, rules, and roles. All right, I want to say a little bit about the mathematics in this course. So if I write an equation like this, dx dt equals minus alpha x, which is like having a concentration of some hormone in the body, let's say, and this is the rate that this hormone is removed. So alpha is units of 1 over time. Some of you might be scratching your head, and some of you might know, oh yeah, the solution is x of t is the initial concentration times e to the minus alpha t. So it's an exponential decay of this concentration because it's removed at a constant rate. How do we know? We can differentiate this, and then the alpha comes in front, and then you have minus alpha x. Some of you, this is like air you breathe. Some of you, you need to freshen up. So I'm aware this is a heterogeneous class. So I want to use each other to help to learn the math. But the level of math we use is going to be stochastic differential equations. On the other hand, when I say something like histone H3K56 acetylation in the subtelomeric region, some of you understand what I said. Other one of you, it's like a foreign language, right? So that's the level of heterogeneity we have in the class. Let's just find out which departments you're from. So what departments are you from? Faculties?
Life sciences.
So life sciences, raise your hand. So it's like 50%. What else?
Physics.
Physics, raise your hand. So I did a PhD in physics. Yeah, great. That's like 10%. What else?
Biophysics.
Biophysics? Yeah. Biophysics. Welcome. Okay. What else?
Computer science.
Computer science. Be proud. So not 10%. What else?
Chemistry.
Chemistry. Raise your hand. It's like 5%. What else?
Mathematics.
Yeah, raise. Okay. Couple. What else? Any MDs? One MD. So we have a doctor in the room. Any engineers? Hi, welcome. Let's take a nice deep sigh of relief so that you can find your way. Any engineers? Other disciplines? Yeah, are you an engineer? What engineer?
Biomedical.
Biomedical engineer. Technio? Anyone else? Yeah?
Computer science and engineering.
Computer science and engineering? What kind of engineering?
What?
computer science engineering?
Yeah.
Great. Yeah. Engineering is a very good background for this. So I don't expect you to know anything, basically, not the math, not the biology. It's my responsibility to teach you. So that's about the math. And now I want you to pair with someone sitting next to you or behind you that's from a different discipline. Introduce yourself. Say your name, where you come from. But everybody has to do that. So maybe behind you. Someone you don't know. Someone you don't know. Okay, thank you. So that's pairing. And now you might be thinking, when is we going to start teaching? So I've already started. I think it's very important to set the frame. I want to also introduce, we have a very special guest today. So at Weizmann we have Sagol Institute for Longevity Research. That's funding a lot of research about longevity. And Sami Sagol is here. So maybe a big round of applause for promoting this field. And Sami, can I ask you something? Can you get up? So with something we think aging is very detrimental. What gets better with age? What gets better with age? Huh? Nothing. Nothing gets better with age. What gets better?
Wisdom.
Wisdom? What does that mean? Like
Perspectives. Let's say 20 years ago, 50 years ago, etc.
Yeah.
Are sitting together.
Yeah.
How many years have you been around the academy?
Like 5 years?
So he's asking how many years have you been around academia? He said 5 years. Yeah.
Me 60. So
60 years. Yeah. So it's actually research shows that the aging brain is wiser brain in different ways. For example, ability to regulate emotions. Does that make sense?
Yes,
Yeah.
absolutely.
And also, although processing speed and memory, short-term memory, long-term memory decline with age, the way decision-making is made changes and uses a lot of real-world knowledge. And so not everything decays with age. And I also, in this course, want to reserve a piece about sociology and humanities of aging. We'll touch about that towards the end. Thank you, Sami.
But I'm not in aging. I'm in rejuvenation.
Sami is a rejuvenated human being. So we'll talk about that too. All right, so let's start with the subject matter. Erase this. It's kind of hot. Is anybody else hot here?
Yeah.
Can we do something about it? Okay, so let's start with trying to define aging, and in that way, we'll also talk about the basic functions that we're going to deal with. So this is basic facts of aging. I want to tell you about quantitative patterns that occur with age, and they're going to be the food for making our mathematical understanding of aging, and also for putting all the biological facts, the thousands of biological facts, in a framework that I think makes it easier to understand. Because otherwise, so many things change with age. It's bewildering. That's part of the problem in this field. But we want to look at it through a direction where things can be understood by a human being. So let's start with what's not aging. So imagine there's a group of organisms, N organisms, living on an island. And they don't age. But there's a predator on the island that eats them at a certain rate. So the eaten at rate HEX, that's called extrinsic mortality, because it comes from the outside. We can plot, this is going to be very important for all the course, what's called the survival curve, which is the fraction surviving as a function of time. So at time t equals 0, let's say they're put on the island, and all of them are surviving, of course. But as time goes by, what's going to happen to them? Their number is going to decline. In fact, it's exactly the same equation. They're eaten at rate h external. So the survival function of t is e to the minus h external t. Starts at 1 and decays. If you look at human beings and you measure the survival curve, human beings tend to die not when they're young, but rather when they're like 70, 80, 90. So this is our hypothetical non-agers, and this is humans, real animals, right? So we think that aging, in one aspect of aging, is that the probability of dying changes with time. When you're young, you don't die. When you're old, you do die. Something changes. It's different when you're 20 and when you're 80. Something in your body is different that affects your probability of dying. Okay. Now, another very useful curve for us is called hazard, which is the probability of dying in a given unit of time. Well, let's say 1 year, in a given year. So if you made it to age 90, what's the probability you'll die between age 90 and 91? That's called mortality or hazard. And so for our non-aging organisms, they're eaten at a constant rate, hx. So their risk of death is basically constant with age, right? Yeah, question.
Sorry to interrupt. For the extrinsic mortality, why is the line not linear, like over time?
So the question is, why doesn't this look like this, a linear line, right? Right? It's because, yeah, the great question. It's because the number of organisms eaten is the number of organisms we have at that time times the rate. So it's an exponential decay, like radioactive particles. Did I answer your question? Sure. Great. So it's our first question. It's not exactly, there was one question, will there be songs? But I love questions, so that's why I use the whiteboard, so I can improvise. Yeah?
Sorry, I haven't done that in a while, but isn't it like a concave curve down rather than convex?
So the question is, isn't this function e to the minus h external t, does it look like this, or like this, or like this, right?
Well, yeah, I mean, just like if there are less of them to choose from, then shouldn't they more steeply decline? No.
You're asking if there's less of them, they might be more steeply declined. Let's say if
they're more likely to be chosen if there are less.
they're more likely to be chosen. So these animals choose, the predators choose at a constant rate. If they chose as a sometimes you have predators that, let's say, if there's a lot of organisms, maybe the predators are afraid. When there are fewer organisms, the predators are more vicious, and then you get something like that. That's certainly possible. But if the organisms are just eating at a constant rate, you get an exponential decline. Let's say, suppose the probability of being eaten is 50% per year. you have 100 organisms, next year you have 50, next year you have 25. Now you see, again, some people here very familiar with math, some people less, brush up. So in this course you'll brush up on math. We have to be, remember if we're feeling like, oh, superior, soon the person who knows biology is going to teach you. Okay, so this first lecture is more quantitative. All right, so, but how does the hazard, and maybe I want to say that the hazard, you can define it mathematically, it's how many organisms die per year, divided by how many organisms there are that year. So this is the equation. It's also the logarithmic derivative of survival. That's why the logarithmic derivative of an exponential is just h external. So these graphs are connected. But now let's draw the fascinating graph of human hazard. So we take everyone born in Sweden in 1900, let's say, and we ask what's the risk. I say Sweden because they have great records. It's like a gold standard for human aging research. And we ask, what's the probability of death at a given age? And look at how this looks for human beings. It starts at about 1 in 1,000 when you're born. Then it drops to 1 in 10,000 around age 10, let's say. And then it rises and plateaus around 10 to the minus 3. And then it starts, now I'm going to try, this is by the way, logarithmic scale. Then it starts rising linearly until around age 40, until around age 80, and then it starts plateauing and reaches a hazard where when you get to 100, your chances of dying in a given year are 50%. So what do you say to someone who's 120?
He hasn't aged over.
Have a nice day. Like when their birthday, right? Say, have a nice day. It's terrible. So we're dealing with morbid issues in this course, but we wanna do something about it. There's a question, yeah.
Would that mean that around age 100 you basically stop aging?
So would that mean that around age 100 you basically stop aging? And because they're similar now to these non-aging organisms, right?
Yeah.
So if it was absolutely a plateau, you can argue yes. I mean, after age 100, like 101, 102, 103, 104, there's something constant. But if you look under the microscope, and that's actually, because data above 100 is so rare, like 1 in 10,000 people make it, but that's growing because of this changing quickly. In fact, we think it's actually is rising, but not exponentially, but rather polynomially there. So there is still aging, and we can try to understand that. Did I answer your question? By the way, the world record is 122, but that's contested. It's not clear if there was, there might be, always there's problems with recording very old ages. But definitely, especially women can make it to 115, 118. Top 10 are all women, a lot of them Japanese women. And so that's like about the human lifespan right now. Now, what I want to say is, what's going on here? So in the early years, the organism develops. So if you look at a toddler, if any of you have ever seen a baby grow, you grow, it's like amazing. Abilities are added. The body is growing. Everything is fresh. It's hard to say that they're beginning to age at age 0. There's a program of growth, development, etc., that ends around age 20, let's say, and then reproduction ability starts at around age, teenage, and peaks around age 25 or something. So this early part is probably not aging. And by the way, this plateau is because of extrinsic mortality. So this is accidents, suicides, homicides. It's more for males than for females. And if you just look at the intrinsic mortality, that's to say deaths that are not from accidents, suicides, or homicides, that it continues like this. And one of the important observations is, I want to know a second color, but I'm challenged by, I want you to also see it. Can you see this? Blue? Back there? Okay. So the hazard, oh, I better use black here. For several decades, rises linearly on this graph, which means, because it's logarithmic graph, exponentially with age. I'll say it another way. Human beings, the risk of dying doubles every 8 years. Okay, so this is and this important law is called Gompertz law. It was discovered in the early 1800s by a mathematician that had work computing life insurance tables. It's very important to know probability to die so you can know how much to charge. And this law is something we'd like to explain. Why does risk of death double every 8 years? Yeah?
Is it similar to other animals?
Yeah, okay. So I say survival, age external, aging, Gompertz, it's universal. So it's nearly universal. So what if you look at survival curve, so of different animals, in the wild, sometimes they're eaten so quickly, so it looks like this. But in a cage, it looks like this. And the Gompertz law applies to mice, flies, worms, even single cells like yeast, monkeys, elephants. It's like, of course, each animal has its own rate. So mice live for 2.5 years. These are, by the way, the famous and favorite model organisms of aging research. We'll talk quite a lot about, in this course, we'll integrate information from model organisms where you can really do experiments that are very powerful, and you can knock out every single gene and see what happens to the lifespan, etc. So mice live 2.5 years, fly lives 30 days, the worm lives 14 days, the yeast, they can divide for, let's say, 20 generations. So we'll talk about other mammals, but there's a wide range of timescales, but the law applies almost universally. But there are animals that don't have that. For example, some trees, the risk of death drops with age. And some animals like hydra, which are these animals that bud and continuously grow, they also don't seem to have age-dependent risk of mortality, or they have negligible senescence. So some animals are quote-unquote models for immortal. There's nothing that says that biological material can't renew itself forever. And perhaps the most famous example is we are alive now, but our ancestors 1 billion years ago made babies. So every time like we have a mother has an egg that's being fertilized, it's rejuvenated, reset, and starts at t = 0. Biological material doesn't necessarily have to age. So we want to understand what's going on here. Yeah.
Do also all those model animals have the plateau as well?
Okay, so the plateau. So do all of these model animals have the plateau? So let's talk about the plateau. So Gompertz law is an exponential, but it doesn't continue forever. All these model animals indeed also slow down. So this is the plateau, or let's say slow down. It's called slow down. It's not really a plateau. It's also a universal feature. Even genetically identical organisms growing in the same conditions have it. So we want to understand that too, what's going on there. Yeah.
It occurred to me, but most people don't live past the age of 80, but the graph goes to 120. How much is the average of the graph, or in terms of this is like a combination of everybody's chances of dying at a given age?
So the question is, your question is that in human beings, the median lifespan, it keeps increasing, but the median lifespan, so this is T50, right? The time where 50% survive. By the way, can you guys in the back still see what I'm writing? Yeah? Okay. So this is median lifespan, right? In human beings right now, it's women is more than men. It's around 80, it depends on the country. This graph of hazard is you take everyone who survived, let's say, till 92 and ask what fraction of them will die until they're 93. And then you take all those that survived to 93 and ask what fraction will die. So the data here is for those people that say would survive to 105 and what fraction of them will die to 106, which is about 50%.
But those people who lived to 105 are shifting the curve for the people who lived when they were who died at 80. No, like their chances of dying were always, or maybe.
Oh, you're asking, are these people special?
Oh, well, are they affecting the graph?
Or are they affecting the graph? Yeah, they're part of the total population.
Yeah.
For sure, they're normalized. Yeah. Okay. So far we talked about one definition of aging, which is some changes that happen, some damage, some changes that happen in the body that make you more fragile and increase your risk of dying with time. And we saw that there's quantitative patterns like exponential risk of dying, called the Gompertz law, and a slowdown. When you look historically, by the way, in Sweden, let's say, the main thing that changed was that there were more deaths at birth and extrinsic mortality was higher, but the slope hardly changed. So we think that the slope, the Gompertz slope, is a biological intrinsic thing, not an extrinsic. So lots of, even now there's countries where there's 1 in 100 births end in the death, either of the mother or of the fetus, of the baby, because of infections and bleeding. With time, this extrinsic mortality goes down, revealing a kind of constant Gompertz law and slowdown. So once you made it past extrinsic mortality, it's like almost doesn't matter where you live. Only very recently has medicine started to shift things for very old people. We'll talk about that. Yeah, there's a question.
I don't know if there's data for other countries, but is it like universal across human countries? The specific
Yeah, so the question is there data for other countries? There's data for many, many countries, and the slope is very, very similar between countries.
So if the numerical value of the alpha is
Doubling every 8 years is very similar between countries. And so within 10%. Okay, so I want to talk about another definition of aging, which is has to do with diseases. So another feature of aging, unfortunately, is that there are certain diseases that you just never get when you're young, thankfully, but you do get when you're old, right? So there's an increase in incidence of a whole class of diseases called age-related diseases. In 1900, people didn't care about them so much because you died from tuberculosis, pneumonia. Now, they're the main killers. And what are these diseases? By the way, everything I'm doing now, we're going to return to in the course and explain. So it's not just a mystery. Why is the Gompertz law? Why is there the slowdown? Why is it nearly universal? We're going to understand. Okay, so this is my promise to you. And we're also going to understand the following fact that age-related disease incidence rises exponentially with age. So age in incidence. What is incidence? You take all the people that don't have the disease at a certain age, 55, say, and you ask how many are going to get it in the next year. It's a little bit like hazard, but for getting a disease, all those who don't have it yet and are alive. So the 4 horsemen, what's called, that kill you in the Western world are, can anybody tell me? Cancer,
Diabetes.
diabetes,
Cardiovascular disease.
cardiovascular like heart attacks, or stroke, and neurodegenerative like Alzheimer. And of course, there's hundreds of others, like cataract, loss of hearing, osteoarthritis. What's your favorite? Okay. So the incidence rises exponentially, reaches a peak, and then goes down. This is, for example, for virtually all cancers, except for very rare cancers of childhood, brain and bone cancer, but almost all cancers do look like this. Okay. And the slope of different, so this is cancer, this is diabetes, this is cardiovascular, and this is neurodegenerative. The slopes are very similar. So again, suggesting that there's some time scale here. That they're not, these diseases are so different. Cancer you get from mutations and inflammation. Neurodegeneration you get from some process in the brain where neurons stop functioning. They're very different diseases, but their statistics are very similar. Why do they decline at very old ages? Everybody who already is susceptible has already gotten it. So if you get to a very old age, you're safe. Centenarians, people who make it to 100, appear to have lots of genes that protect you from the risk of these diseases. You dealt a good hand. Shaked.
I would expect that if we consider evolution, so we'd say like, I don't know, a plateau until the age of 20 or 30, and only then the graphs will begin to increase.
Okay, so Shaked says, I would consider, on the diseases, for example,
Both?
or both, that if you consider evolution, you would have a plateau in reproductive ages, right? That's what you meant?
Yeah.
And then a rise after that. Why would you expect that based on evolution?
I don't know if plateau, but like, well, if we believe that evolution like motivates biology,
Yeah.
so we are only good until the age of 20 or 30, so people who...
We're only good until the age of 20 or 30. So why do we live till 80? Yeah, it's amazing, right? So there's a whole section on evolutionary theories of aging that we'll get to. I'll get to it in a second. But that's a very good way of thinking. So after reproduction, of course, human females can't reproduce after a certain age, let's say 52, 53, 55, something like that. But they live to 80, 90. So people say after reproduction, you're in the shadow of natural selection, mutations that make you live longer. Won't be passed to the next generation. But there's different ways to think about it. For example, the contribution of old individuals to the 50% of the genes that are in their kids. So that's one mechanism called the grandmother theory. You love it? You're so happy. You worked on that? A few years ago. Yeah, but it's we'll talk a lot about theory. I mean, there's different theories. It's very hard to test. But there are experiments you can do, etc. Okay. So we want to understand why are these slopes so similar, and what's going on here? I mean, with age-related diseases. But I also want to say that a motivation for the field of longevity, one motivation is to say that aging is the main risk factor for these diseases. So, for example, chance of getting cancer when you're 70 versus 20 is like 4,000 times higher. It's much more than smoking. It's much more than sunlight. It's the main risk factor. Even though I want to tell you, one of my pet peeves, that all the cancer labs here work on young mice when they study cancer. It's like there's a disconnect in general between the field of cancer, field of aging that I want to address. Where's the Owen? Write that down. So it's the main risk factor. So one of the ways of thinking is that medicine today treats each disease once you get it, cancer, diabetes. If you can slow down aging or something like that, you address all of the diseases in one fell swoop, right? You delay them. And some people say, why study aging? I mean, do you want more people in the world? Climate change? So in fact, the economics of it is that if you consider it's not only that you have less healthcare costs if you improve health of old people, they're more productive, and that's a much higher economic benefit. So it's like a huge amount of additional wisdom cross experience, cross ability to function in the world, and that can be unleashed. It's probably not so easy to extend the human lifespan, but to compress the sick span is a big goal of longevity research. Did I explain myself? So I think it's ethical to study longevity from the point of view of things like climate change. Okay, welcome back from the break. I had some questions in the break. So one thing is about if you're not enrolled in the course, if you want to be enrolled in the course but you missed the deadline or something, write me an email, or Ben here has his email, or me, and we'll get you enlisted, enrolled, not enlisted. It's a different thing. There's a question about, I mentioned the grandmother theory, right, which is actually not a very strong theory. It's that grandmothers can contribute to the survival of their genetically related kin. But then some questions were, what about flies and worms, right? Are their grandmothers also taking care of their kin? So how can this be universal, etc.? So that's a superb question. Why is what's going on here with the universality across so different organisms? And I also mentioned that there was a recent paper suggesting that old worms, they can't move anymore, but they're still alive, so they're fresh, and then when they die, they're cannibalized by the young. So that's another kind of grandmother effect.
From the inside.
I'd not recommended. Don't eat your grandmother. But it's very complicated, ecology. But anyway, that's kind of mysteries one explain. How can something be universal across so many different kinds of biology, and what's the molecular mechanisms? And we'll take our course to do that, and we'll also devote time to evolutionary thinking. Yeah.
So could it also be maybe a function of how much society and population has or species has? Like if there's a lot of like societal structure or something, then you sort of have this grandmother effect.
Okay. The question is, if there's more social structure, is there more grandmother effect? There's more longevity. Great research question. Let's talk about data that can address that, because what I want to go now, so I did disease incidence, and I want to, since you guys are so fascinated, I want to make a little bit of a description about what happens with different animals in terms of aging, different mammals, and then I want to talk about another definition of aging, not only increased rate of dying and increased risk of disease, but continuous decline in performance. So we'll talk about that too. So let me start with what's called comparative aging, or comparing different mammals. So, before that, let's take a nice deep sigh of relief. So if you look at different mammals, you can plot their mass, say log mass. So you have mice and shrews that are 2 grams, or let's say 1 gram. And you can have whales that are 10 to the 8 grams, which is 100 tons. That's mammals, right? And you think about their lifespan. So this is going to be the log lifespan, which means the longest lived individual in the zoo or known to science. And there's data for about 1,500 different mammalian species. So for example, if we think about the mouse, it lives for about 2.5 years, weighs about 1 gram. If we think about the bow-headed whale, it lives about 200 years. This is the bow-headed whale, weighs about 100 tons. And so let's say this is like, 1, 10, 100, 1,000. It's generally observed that the bigger the animal, the longer it lives. Right? So if you take, let's say, a dog that weighs 10 kilos, maybe lives for 15 years or something, and you have, say, dog, and you have cow, and you have elephant, and you have hamsters. What weighs 100 grams?
Awesome.
Huh? Awesome. Shrew. So it used to be thought that there's a kind of a relationship where longevity is, it goes kind of like mass to the one square root of mass. So if you're here, you're 1 gram, 10 to the 8 gram. So 1/4 power of 10 to the 8 gram is 100. So if a mouse lives 2 years, the whale lives 200 years. But that is an example of what's called cherry picking, because in fact there are animals that are small and live long. For example, bats, many species of bats, they're like mice that live not 200 years, but maybe 40 years, something like that. And people are also, how much do we weigh? 10 to the kilo is 10 to the 3rd gram. 10 to the 5th? Yeah. So somewhere here. And in fact, and this is something that we did in our lab in 2016. We took all this data and we observed that it fits inside a wedge that we called the mass longevity triangle. And what's the idea? So you don't have animals that are small, like weigh 10 grams and live, weigh tons and live a couple of years. You don't have that. Right? So the basic idea in evolutionary theory of aging is it's the extrinsic mortality that determines your lifespan. So if you're a mouse and you know that you're going to be eaten in 1 year, it doesn't pay to repair yourself to live 100 years. That resources you can invest in growing quickly and making lots of babies before you're eaten by the owl. Did I explain myself? That kind of trade-off theory, sometimes called disposable soma theory. So the extrinsic mortality, which determines the trade-off between reproduction and growth versus repair. So if you're killed often, you go for you want to grow quickly and reproduce. Many babies, fast, cheap, and out of control, like mice. But if you're so big, like a whale or an elephant, you're not killed that often. Do you agree? So you grow very slowly, and you make one baby, and you care for it for lots of years. You make better quality offspring, quote unquote. This raised the question in your mind. Huh?
Several questions.
Okay, good.
If we compare, prey versus like meat eaters versus grass eaters.
So the question is about carnivores versus herbivores. Yeah, it's not a huge effect here.
Yeah, and one of them is being eaten more than the other.
Yeah. So what about the bats? So the idea is that if you have a triangle shape, that's a theory in evolutionary theory. When you have a trade-off, you can say that this is like archetype strategy for what you might call fast reproduction and growth, right? So this is high extrinsic mortality, rapid reproduction and growth. This is you're big, so you can you have low. You're protected. You're big, so you're protected. And these animals are very interesting. They're small, but they live in a protected niche. So bats can fly. And if you look at the other animals near here, it's like a protected niche. If you look at the other animals near here, you find rodents that live underground. They're called naked mole rats. They live for a long time. So they're underground, and they're very social, by the way. There's blind mole rat and naked mole rat. One is more social than the other. But they live much longer than mice, like decades. Or primates also. Primates, we call it the cognitive niche. The primates are here. So if you look at birds, the flying birds are here and flightless birds are here. They're not mammals, but they have a similar design. So in evolutionary theory, if you have three possible strategies, three archetypes, you expect that biology will fill the triangle where these archetypes are the vertices. The reason is that for every point in the triangle, right, every point outside of the triangle, if there was an animal here, there's a point inside the triangle that's closer to all three vertices. It means it's better at all three tasks. So you can erase this point, and you're left with a triangle. So if you see a triangle, you know there's three tasks. If you see a tetrahedron, you know there's four tasks. That's like a kind of evolutionary theory of optimality that you can read about in this paper. So that's what data looks like for mammals and for birds. And
what's the upper strategy seems to be the upper left seems to be the lower one, but executed better.
this one?
Yeah.
So protected niche. So if you look at bats and compare them to mice, mice make a lot of babies. They say 8 babies every 3 months. Bats make one baby and care for it for taking it on their back and take care of it for a year or two, show it where the fruit tree is 40 kilometers away. Completely different strategy. Also, the ones near the protected niche have a larger brain to body size ratio, so they're smarter, you can say. So
So it seems to be the same strategy as the bigger ones, just with smaller size.
it's more similar to this strategy here, but more extreme in terms of slow development and taking care of children even. Because I think to live in a protected niche, you have to teach how to live in the niche. Think about human beings. There's no other primate that has a childhood, this period. They go like baby, toddler, teenager. They don't have the childhood where you grow 6 centimeters a year and you learn society, basically. You're in 6th grade and you learn how to fight for Markata Kita and so they don't have that. So we have anyway. This is my kids are teenagers. I have 3 teenagers. So I can tell you that. I can tell you many things, and they helped me write the song. So remember. All right. So that's a taste because we're going to have a lecture about comparative aging, and evolution of aging. And stuff like that.
Is this only for mammals? Like, if I take sea turtles, which have lots of offspring, can live longer, but have lots of things that it's
Yeah. So the question is about what happens when it's not mammals, like turtles. This is mammals, yeah. But I'm not aware if this analysis was done for reptiles or for invertebrates, which might have different constraints. Yeah. Okay. I'm giving you here some basic facts of aging. and I want to get to some juicy stuff. So I think you guys like evolutionary theories of aging, so I'm going to make sure to teach that. Another quantitative fact of aging. So we saw risk of death rises with age, and it kind of rises exponentially with age. And risk of disease rises exponentially with age. Exponential is very sudden, right? You're not sick, and then you're sick, or you're not dying, and then you're dying, right? If you've seen very old people, at some point they start losing function very quickly. So it's like, but there's something that also is very gradual about aging, and that's when you measure not diseases but performance, like your ability of your kidney to filter the blood, or your grip strength, or how much VO2 max, how much oxygen you, when you're like running as fast as you can, how much oxygen your body can take in. These are measurables that we call performances. And unlike these exponential things, performance generally decays linearly with age. So it like, looks like this. This is age. And now let's talk about, for example, muscle strength. So let's imagine that at age 20, it's 100%. Now, when you look at muscle strength, different individuals can be very different. If you're exercising, you're not exercising, right? So I'm going to start with cross-sectional data, which is how in the 1950s people started studying this. And that means you take all the population that's 20, 30, 40, 80, and you do the average. So 100% will be the average of our overall 20-year-olds. And then you recruit some 30-year-olds, 4-year. And what you see is, and I'm going to write here, 0% is actually the minimum needed to stay alive in bed, let's call it like that. It's the basal level without any challenges. So in general, they decline like this on the cross-sectional level. Like that. And if I write down here kidney filtration rate, what's called glomerular filtration rate, it follows a very similar slope when you normalize it. So, of course, each one has different units, etc., and each one has a different basal level. But when you start from 100 and you say 0 is the basal level, they collapse on a linear curve. On the when you do cross-sectional studies, that's the average all the population. Which when physicists see data collapse, they get very excited. Again, so there's something uniting these very different functions. Am I conveying my enthusiasm? So when you look, so this is like, when you look at what's called longitudinal, which is this, you follow a single individual over time, then people can be very different, and they can, let's say this person compared to the population, it starts lower, and they can decline like that. Or so this person is like a world-class athlete, and they decline like that. So the thing is that when people die, you remove them from the sample, and the cross-sectional data looks linear. So demographics is very subtle. So you have to be, that's something maybe we'll discuss statistically, the difference between following individuals over time and averaging at a same time point when you're losing individuals all the time. So this is more like the sudden collapse that we see with others. But still really interesting to think about this population linear decline. By the way, when does it hit 0? Anybody want to guess? So So if you extrapolate out, it hits zero at around 110, which is like close to the maximal lifespan. So that's also interesting. Questions? By the way, yeah, go ahead.
Is most of the decline from the longitudinal decline or from the collapses?
Yeah, so longitudinally, if you think about, I don't know if you're old enough to already notice this, but longitudinally, each of us notices this thing. And it's very gradual, and then there's a change like that. So the linear thing applies to everyone until a certain...
So is the linear slope of each individual similar to the cross-sectional one? Or is it more influenced by the collapses?
No, this could be variable. So for instance, if you're exercising, you can maintain your muscle strength more. And it has to do with lifestyle, genetics. So that's performance. By the way, if you do cognitive performance, there's no minimal basal level for cognitive performance, but you do like a z-score, which means how well you do on a test compared to average or something. So a lot of cognitive things, again, they look like this. For example, memory, long-term and short-term memory, and also computational speed. But like I said, when Sami was, I forced Sami to talk, other things improve with age, especially what's called world knowledge or crystallized knowledge. ידע כללי. All right, so that's another. So you can say that aging is kind of a gradual, then sudden decline in abilities. Okay. What can we do about aging? Here I want to switch gears to see what aging longevity researchers are doing, which is looking for ways to slow down aging and affect these graphs. So I want to tell you about that. There was a question?
Yeah, maybe you might answer, but is it ever possible to recover from the steep decline? Like, I imagine if you start working out while you're still in like the linear portion, maybe it will improve.
Oh,
But, like,
the question is,
the steep decline-
if you're in the steep decline part and you start working out, can you improve? Yeah. I don't know in humans, but in animals, like worms, yes. I'll tell you about that, and it's like, that's actually quite fascinating. Okay, listen. So I'm going to now talk about longevity interventions and additional interesting patterns. Let's first take a nice deep sigh of relief. One of the big important things, by the way, is called VO2 max, which is how much oxygen you can assimilate in your body when you're running on a treadmill and your heart is beating very fast. It's really important, and doctors say that below the neck, it's the one number they most trust to know how likely you are to die, because when you're at the end of life and there's something very bad happening to you, people breathe very, very strong in order to get the energy and the senses to deal with whatever it is. And the more your VO2 max, the more spare capacity you have, for example. So that's one of the big things that also declines. Athletes decline. So I wanted to talk about longevity interventions. So this field of longevity interventions, it's not clear for human beings, but it's quite clear for the different animals that we talk about, the yeast, flies, worms, mice. Right now, it's a phase where clinical trials have begun in humans of some of the drugs and interventions that were found in animals, but it's not clear. It's not clear if you can extend the maximum human lifespan, or we don't know yet. So it's up to you guys to think. Maybe one of you will discover the drug.
So loud.
So the typical curve is, again, the survival curve that I started out with. Age, survival, a fraction of organisms surviving. And when you do an experiment, let's say on, most of what we know comes from simple organisms. The simpler the organism, the more experimental power you have. So a lot of things were discovered in yeast, worms, and flies. And then were tested in mice. And sometimes in monkeys. Now also dogs with their owners, pet dogs. So if the owners want the dog to live longer. Oh, I forgot to tell you something. What lives longer? A mouse or an elephant? What lives longer? A small yippy dog or a big dog? What's going on?
More cells.
Huh?
More cells, save genes.
So when you look between species, bigger lives longer. Within a species, it's the opposite. And that has to do with breeding for specific mutations. They're like longevity mutations. I'll talk about it. If you have a corgi and in my family, there's a corgi and shepherd in the same family. So, all right, so so we have our T50, which is the age of 50% survival, 0.5. And now what we're looking for is doing something to the organism that will extend T50. Right? That's the golden graph you look for in this field, experimental field. And so what can do that? So there's quite a lot of different things that can do that. So especially short-lived animals like worms that live 14 days, you can triple their lifespan or more. Mouse, you can probably extend it by 40%. So I just want to say, mouse, you can extend it in the laboratory by 40%. But evolution, starting about 80 million years ago with the ancestor of all mammals, extended it by 100 times. So what we can do in the lab is very meager compared to what natural selection can do. Why is that? What's going on? Okay. So longevity intervention. So, for example, one thing that seems to work in all these animals is called caloric restriction, which is to reduce calories by 30 to 40%. Not recommended because you lose a lot of weight and muscle mass and become very fragile. So it's good in a cage, but if you're in the real world, it's not so recommended. It also feels terrible. But it extends lifespan. Also, all kind of intermittent fasting and reducing components of the diet, ketogenic diet, reducing methionine, etc. What's going on here is thought to be, do you remember I told you that there was a balance between grow and reproduce versus repair damage? So it turns out that when you're starved, cells in our body delay growth and reproduction until there's food around. And instead, they recycle and reduce. And so this caloric restriction pushes you towards here, which is great. It is great for fitness, for reproductive fitness, because if you don't have, like a worm that says no food now, I'm not going to lay my eggs now. I'm going to repair myself to last another few days till there's food. That makes sense, right? So that's how it could be the reproduction is linked to, it's not selected for long life, it's like a program that. Can you close the door? So caloric restriction does that, and then there's different mutations and stuff like that appear to work similar to caloric restriction. So there's something called IGF-1 pathway, which IGF-1 pushes towards growth and reproduction. So if you delete or decrease IGF-1, you live longer. It's like mimicking passing. And even human beings that have mutations in this hormonal pathway, they're very short dwarfs, but they have less cancer, diabetes, and stuff like that. So there's very conserved. And this also explains the dogs. So when there was a selection of dogs, breeding dogs for bigger or smaller, it played with this pathway, made them grow a bit fast, big, at the expense of repair. So dog breeding goes against, so evolution doesn't do that. It doesn't use this pathway for longevity. It doesn't. What does it do? We'll talk about it, but the long-lived animals have better repair, like better double-stranded break repair and things like that. They have some commonalities that we can learn from, maybe. We learn from what whales do. So that's an example. And then there's a whole bunch of additional things. I'll get into like rapamycin, which is like a drug that also mimics caloric restriction, and many, many, many other things that we'll talk about at length. But I want to talk about quantitative patterns here. This has to do with the paper that got me into the field of aging. So as I said, I did a PhD in physics about turbulence, and I moved to biology in my postdoc, and I studied gene regulation networks inside cells. And at some point, and I thought that aging and things like that is like just decay, entropy, not interesting, too complicated. But then I saw a paper that showed a data collapse, and the paper said, let's look at these longevity interventions, right? And ask, they extend the median lifespan, but what do they do to the shape of the curve? So if you plot the same curves, but normalize by the median lifespan, so now you reach 50% survival at 1, right? This is, so if the caloric restriction is green, you can't see the green, it's blue, it falls on the same curve. So that means you extend life, but you preserve the shape of this. It's like there's one time scale that you're stretching. And when I saw that, I said, wow, there's something to understand here. So this is called scaling. This is the paper. Actually, it's the same year. So what's going on here? By the way, there are a few interventions that steepen, and they're going to be very interesting for us because they change something else, not only living slowly and quickly.
You said that longevity research makes you live more healthy life, not like longer life. How does it make sense here that like it seems as also unhealthy people live longer as well? Like
Here?
yeah, when you like give the
Oh, the caloric restriction?
Yeah.
So what I want to say is that in mice, it's possible to extend not only the median lifespan, but also the maximum lifespan. Like I said, by 40%, something like that. 10, 20, 30, 40, maybe 50. In human beings, it's not clear if you can do that. And therefore, the focus is on, as I'll tell you, is more on reducing the fraction of time when you're sick. But it might be possible to extend a little bit. It's not known right now. Maybe a breakthrough will help you extend by a lot, but it's not clear. But you're asking, these animals, they're not sick, they're just like very thin. And they're living in cages, so they don't have to deal with the real world. So, but with rapamycin, they look fine, and they also, at a given age, they're healthier. So definitely you can also extend the healthy part of mice lifespan. That's for sure. The last point I want to make, I think we could, it's a lot of information, I want to acknowledge that. Is a fascinating question of what happens when you do an intervention that extends life, but you do it when in the middle of life, not from the beginning. Can you forgive the sins of the past? This was a little bit about the question, what happens when you're old and you do the intervention? So this is experiment on fruit flies, where you can. Fruit flies are good because you can follow millions of fruit flies' lives and deaths, which are quite helpful organisms. So you can look at hazard curves, and like I told you, there's a Gompertz law and a slowdown. And this here is the curve of flies on a better diet, on a longevity diet. So they die at every age. They die less. Does this make sense? Now the question is, what happens when you? So this is normal, like normal diet. The question is, what happens where in middle you shift from normal to better? So the answer is that you do this. There's a rapid shift. Also here. Yeah.
Is there a limit to when you can do this?
Is there a limit to when you can do this? I know an experiment in worms where they let 75% of the worms die, so that's a very old age, and then they added the genetically turned off this IGF-1 pathway, and they got almost the full lifespan extension for those very old worms. But they looked very bad. So old worms have cracked cuticles. A lot of functions stayed there, but some functions improved, and those give you clues to what are the drivers of aging. Did that answer your question?
Yeah.
So it's like it's not like you suddenly become like you look like you're 20 years old, but there are other things you can do, like change of temperature, that you don't forget the sins of the past, and you just change the slope. So it depends on the intervention. But what this does tell you, scaling tells you there's one like long time scale in aging, and here it says there's also a short time scale, a quick one, that we need to understand. And you know, the big thing that's staring at us from the whiteboard is the fact that all these flies and these worms, they're genetically identical, and they're raised in the same conditions. But you see that they're dying at different times because it's not the survival curve. It goes. So an amazing and kind of unexplained thing is that aging is random, it was stochastic, but it's not, the stochastic, it's not genetic, and it's not environment, it's something else. Right? Why? Because you take genetically identical mice, grow them in the same conditions, they die at different times. Take monozygotic twins, human beings with the same DNA, grown in the same family, and they die at different times, almost like two unrelated people. The correlation between their death times is 0.2. So it's not genetic, it's not environmental. What is it? Something else we want to address.
Stochasticity isn't genetic.
Yeah, it's something stochastic. That's really interesting too. And also universal. All these things are universal, nearly universal facts of age.
Could it be epigenetics?
Yeah, it could be epigenetics.
Aging stochastic or maybe death?
Death is stochastic. By the way, what you die of is very genetic. Like if you die of cancer or diabetes, that's affected, let's say, some diseases it could be even 70% heritable. What you die of is very affected by genes, environment, but your lifespan is less. So that we also devote time to that, the genetics and the heritability of aging. And this lecture was very quantitative, let's say, graphs and stuff, but we'll also talk about particular biological pathways that go wrong in this course. And in fact, one of our major goals in this course is to understand what is the in each of these animals, what is the main driver of aging biologically, and maybe that's a target for addressing aging. Okay, I think we can stop earlier today. What do you think? Let's take a nice deep sigh of relief. Before you go, very important announcement. For reasons beyond my control, I won't be here in the next 2 weeks. I have to. So we're going to have next week no lecture. Week after that, Ben is going to give a lecture preparing you for the exercise. And so in 3 weeks we continue for where we started. I apologize for that. Okay, let's take a nice deep sigh of relief. Thank you.
Thank you.